We will now talk about development of the heart. Remember, this is embryonic development of the heart. Before we start talking about cardiogenesis, maybe I just need to tell you that development of the heart is one of the complex things in embryology. I've tried to simplify it, or at least to just bring out the components which I think are necessary for a BSc nursing to know. Otherwise, there's a lot about development of the heart that I'll not talk about. There are books and books concerning with cardiovascular development. In this lecture, we are going to state why the cardiovascular system must develop very early during embryonic period. We are also going to state the unique features of the fetal cardiovascular system. This is if we compare it with the adult type or other the postnatal type of the cardiovascular system. We will talk about two processes, angiogenesis and hematogenesis. I'll define what they are and we'll talk about them. The structure that becomes the heart is initially just one tube. We call the primitive heart tube. We are going to see how that primitive heart tube is formed. That primitive heart tube has different segments. So those different segments, we are going to see what they become. For them to become the things they become, those segments must undergo septation so that you have 4 chambers. And so we're going to see the different types of septations in the heart. So the first agenda is, why should the cardiovascular system be the first one to develop that early? Yes, I've already told you that the cardiovascular system is the first system to fully function in the embryo. As a matter of fact, the first heartbeat that beat spontaneously, it was or is experienced in the 4th week of gestation. And from that time onwards, your heart has never stopped beating. The heart starts to beat in the 4th week of gestation, very early in fetal life. And why is that so? You know, the fetus, the embryo and the fetus growing so rapidly, and that means that there's demand for nutrients, there's demand for oxygen, demand for hormones, and there's need even to transport substances. So we need an efficient way in which these substances can be transported. Diffusion alone cannot meet this demand. If the tissues are growing so fast, diffusion alone cannot meet the demand of transporting substances. And that's why the cardiovascular system develops so early in embryonic life. Our second objective is to talk about the unique features of the fetal cardiovascular system. If we compare the fetal cardiovascular system with the cardiovascular system in the adult, there's some 3 things that are important to highlight. One of them is that the fetal cardiovascular system does not have a functional pulmonary circulation. This is to mean that blood does not go to the fetal lung for the purpose of gas exchange. Of course, it does that in the postnatal type, but not in the fetal type. And tailored to that, we know that the fetal cardiovascular system has a placental circulation. This is where gas exchange takes place. You and I don't have a placental circulation, even if you wish. And the third one is also very important. There'll be oxygenated streams of blood and deoxygenated streams of blood. In the postnatal type, those two streams of blood never mix. However, in the fetal cardiovascular system, the oxygenated stream of blood mixes with the deoxygenated stream of blood. So those three things are important to note about the fetal cardiovascular system. And so to achieve these things, there's a particular way that the fetal cardiovascular system is designed. From an anatomical point of view, we can say the following: that the fetal cardiovascular system has vessels that connect it to and from the placenta. So those vessels will then maintain continuous blood flow to and from the placenta. That is a placental circulation. Those vessels are the umbilical arteries and the umbilical vein. Also, because we say that we don't have a functional pulmonary circulation, the fetal cardiovascular system is designed in such a way that it shunts. To shunt is to skip. It shunts the pulmonary circulation so that blood does not go to the lungs, or rather a lot of blood will not go to the lung. So there's a way blood that is supposed to go to the lung is redirected away from the lungs. The third thing to note is that despite the fact that we've said that oxygenated blood mixes with deoxygenated stream of blood, the fetal brain really requires a lot of oxygen. And so despite mixing of oxygenated with deoxygenated streams of blood, there's a way the fetal cardiovascular system is designed so that a relatively oxygenated stream of blood actually goes to the brain, even though oxygenated and deoxygenated streams mix eventually. There's a way oxygen is redirected to the brain. And lastly, you realize that those things which you've mentioned are very, very unique and fundamental for the fetal cardiovascular system. Yet at the time of birth, they all stop. There's a radical change that occurs at birth. Fetal cardiovascular system is designed in such a way that it can anticipate and handle those radical changes that will be taking place at birth. That is what is unique about the fetal cardiovascular system. Now we are going to talk about angiogenesis and hematogenesis. Angiogenesis is the process of formation of blood vessels. How do blood vessels form? Blood vessels form from mesenchyme. The mesenchymal cells differentiate to become what we call angioblasts. Angioblasts are vessel-forming cells. So if you take this to be mesenchymal cells, they differentiate to become angioblasts. Angioblasts then regroup together to form what we call blood islands. So you can assume these are grouping of angioblasts. The grouping together of mesenchymal of angioblasts is what we call the blood islands. Of course, they'll group in such a way that they're cord-like structures. It's not just a ball, but more of a cord-like structure, branching or not branching, either way. Now, if you're going to form blood vessels, then this is what happens. In any blood island, there'll be those cells in the center and the cells in the periphery. If you're going to form a blood vessel, then the cells in the periphery are the ones that flatten. Even though they flatten, as shown here, those cells retain their intercellular junctions. They retain their intercellular contacts. So therefore, that will constitute the endothelial lining of blood vessel, which is basically a capillary. That's how blood vessels form. And how do blood cells form? The process of formation of blood cells is what we are calling hematogenesis. You can call it hemopoiesis, but let's call it hematogenesis. So hematogenesis is the process of formation of blood cells. And how do blood cells form? For blood cells, we'll now focus on the cells at the center of the blood island. The cells at the center of the blood island detach from each other, which means they become free. So they'll not behave like the cells in the periphery of an island, which retain their contacts. The cells at the center become free, and they're the ones that will differentiate into the different types of blood cells. In terms of sites of hematogenesis, there are three phases based on site. The yolk sac phase of hematogenesis is that period when blood cells are forming within the yolk sac as well as the chorion. We call that the yolk sac phase of hematogenesis. We also have the hepatosplenic phase of hematogenesis. The hepatosplenic phase of hematogenesis is when blood cells are forming within the liver, spleen, and lymph nodes. Those three sites constitute the hepatosplenic phase of hematogenesis. The last phase of hematogenesis is what you're calling the medullary phase of hematogenesis. Medullary phase of hematogenesis is when blood cells form within the bone marrow. So this is what will begin yolk sac phase. It will be followed by hepatosplenic phase. Towards the time of delivery, the medullary phase is the one that will predominate. Postnatally, the normal site of hematogenesis is bone marrow. The liver and spleen are not expected to form blood postnatally in as much as they do prenatally. That does not mean that the liver and the spleen don't have the capacity to form blood. As a matter of fact, sometimes there are some diseases that infiltrate the bone marrow, and so the bone marrow fails. If the bone marrow fails, then the liver and the spleen are recruited to form blood. And at that instance, we call that extramedullary hemopoiesis or extramedullary hematogenesis. Formation of blood outside bone marrow. That's a term you use largely postnatally. Okay, so we now want to see how the primitive heart tube is formed. The primitive heart tube is that tube that will become the heart. How is it formed initially? We've agreed that for you to form blood vessels, the cells in the periphery of the blood islands are the ones that will attach together, flatten out and attach together to form blood vessels. If that vessel is falling at the region where the heart is supposed to be, then we will not call that a blood vessel. We will call that an endocardial tube. So look at this. We have angioblastic cells which form blood islands. Those blood islands then eventually form something which is tubular, which will be like a blood vessel. But if that blood vessel is falling at the region where we expect the future heart, then we'll not call it a blood vessel. We call it endocardial tube. There are two endocardial tubes, the right and the left endocardial tube. These two endocardial tubes fuse together when the embryo falls in the transverse axis. Transverse embryonic folding leads to the fusion of the right and the left endocardial tube. When the right and the left endocardial tube fuses, they form the primitive heart tube. So the primitive heart tube is formed from the fusion of the right and the left endocardial tube. That is how the primitive heart tube is formed. Let's take this to be the endocardial tubes, right and left. As the embryo falls in the transverse manner, as we can see here, endocardial tubes, the two endocardial tubes fuse to form the primitive heart tube, which you now see here. Okay, what are the parts and derivatives of the primitive heart tube? So this is the primitive heart tube still, the right and the left endocardial tube coming together to form the primitive heart tube. This primitive heart tube develops some chambers which are in series. You realize it's one lumen at this point in time. It develops some chambers which are in series. And let's use this image to describe those chambers. So blood comes through this end and that end. And this is the endocardial, this is the primitive heart tube, the fused parts of the two. The unfused parts are not necessarily part of the primitive heart tube. Blood flows through this end and comes out through that end. And that end and comes out through this end. So this common chamber is the primitive heart tube. The primitive heart tube has 4 segments. This region here, where the 2 vessels are meeting, is what we call the sinus venosus. From the sinus venosus, blood flows to the region we call the primitive atrium. From the primitive atrium, blood flows to what we call the primitive ventricle. And from the primitive ventricle, blood flows to this big region, which we call the bulbous cordis. The bulbous cordis has 2 components. The proximal component of the bulbous cordis is what we call the conus arteriosus. The distal component of the bulbous cordis is what we call the truncus arteriosus. This is conus arteriosus. This is truncus arteriosus. Those are the chambers or the parts of the primitive heart tube. From this primitive heart tube, those chambers, those segments of the primitive heart tube are going to give us the various chambers of the heart. So let's see the derivatives of these different parts of the primitive heart tube. And we start with the sinus venosus. This is the region of the sinus venosus. What does sinus venosus become? In adult, the sinus venosus forms the smooth part of the right atrium. The smooth part of the right atrium is known as the sinus venarum. I showed you some inner surface of the heart, and I told you that you can have a smooth part and a trabeculated part. The trabeculated part is the rough part. So right atrium has a smooth part and a rough part. The smooth part is what we're calling sinus venarum. That smooth part comes from the sinus venosus. The other structure that comes from sinus venosus is the coronary sinus. Remember, coronary sinus is the largest vein that drains the wall of the heart. These 2 are the derivatives of the sinus venosus. Then now, blood from the sinus venosus goes to the primitive atrium. What are the derivatives of the primitive atrium? The primitive atrium divides. There'll be a septum that forms in the middle of the primitive atrium. And so with that in mind, after the primitive atrium has undergone septation, which means formation of a septum, it will divide, that septum will divide the primitive atrium into 2, the right and the left atrium. But which part of the right atrium will we form? We will form now the rough part of the right atrium. The rough part of the right atrium is what we are calling musculi pectinati. the pectinate muscle. You can just call it the trabeculated part of the right atrium. It also forms the left atrium. So the other chamber after that septum will be the left atrium. Okay, so from the atria, we go to the ventricles. This is the primitive ventricle. The primitive ventricle also undergoes septation. In this manner, there'll be a septum that forms here. That septum divides the primitive ventricle also into two. It will divide that septum, which we are calling interventricular septum, will divide the primitive ventricle into two parts. It will divide it into the trabeculated parts of the right and the left ventricle. If you remember from the image I showed you, the trabeculated part of the ventricles were the lower parts of the ventricles. So basically, the primitive ventricle gives you the lower parts of the right and the left ventricle. And that means that you then expect to have the upper parts of the right and the left ventricle as well. Now look at this one. This is the primitive ventricle. This is the bulbus cordis, which has two components, the conus arteriosus and the truncus arteriosus. So the conus, together with the primitive ventricle, are the ones which participate in formation of the right and the left ventricles. Conus, together with the primitive ventricle, they're the ones that participate in the formation of the right and left ventricle. So they'll undergo septation. So primitive ventricle particularly gives you the rough part of the two ventricles. And that means that conus arteriosus will give you the smooth part of the two ventricles. But let's talk about that in its own entity. The bulbus cordis, we agreed, has two parts. Conus arteriosus proximally and truncus arteriosus distally. The bulbus cordis undergoes septation as well. Because we are splitting the conus together with the truncus, we can just call that conotruncal septation. The conus arteriosus component of the bulbus cordis will give you the smooth parts of the right and left ventricles. The truncus arteriosus component of the bulbus cordis gives rise to the root of the major arteries. The root of the major arteries here being the ascending aorta and the pulmonary trunk. So we'll expect the pulmonary trunk to be arising from the right ventricle and the ascending aorta arising from the left ventricle. Those are the derivatives of the primitive heart tube. I now want to talk about the different type of septations in the heart so that we get those four chambers of the heart. There are four types of cardiac septation. We have what we call atrioventricular septation, interatrial septation, interventricular septation, and conotruncal septation. When you talk of atrioventricular septation, we mean we want to separate the atria from the ventricles. When you talk about interatrial septation, we mean you want to separate the right atrium from the right ventricle using that septum. When we talk about interventricular septation, it means you want to separate the right ventricle from the left ventricle using a septum here. And when you talk of conotruncal septation, it means you want to separate the upper parts of the two ventricles together with the outflow tracts from the heart, which are pulmonary trunk and the aorta. Conotruncal septation. Let's see how those different types of septations take place. We begin with atrioventricular septation. For you to separate the atria and the ventricles, you don't need to have it complete septum. Atrioventricular septation is a partial separation of the primitive atria from the primitive ventricle. How does this separation occur? It occurs by development of some tissues which we call endocardial cushions. The endocardial cushions grow towards each other and meet in the midline. So if this is the region of the primitive atrium and this is the region of the primitive ventricle, the endocardial cushions grow anteroposteriorly and meet in the midline. If you take that section in black and look at it from a coronal view, then to look like this. These endocardial cushions just grow in the midline, and therefore we have an anterior and posterior endocardial cushion that meet in the midline, which is this fused part. With that in mind, then you realize that you form two canals on either side of the endocardial cushion. Those two canals are the ones we are calling atrioventricular canal. The right and the left atrioventricular canal will form the future right and left atrioventricular valves. So we need the canals to be there. Therefore, blood enters through the right atrium like that and flows through the right as well as the left atrioventricular canal. That is atrioventricular septation. I want to ask a question with regard to that concept of atrioventricular septation, and I want you to answer. I want you to answer in the chat. I have enabled the chat to be public for everyone. So the question I'm asking you is, if atrioventricular septation goes abnormally, what kind of congenital heart diseases would you expect to happen with regard to abnormal atrioventricular septation? Just write so that everyone can see, so that you don't have to repeat what someone else has written. You can write in the chat, I'll be seeing. What kind of congenital anomalies do you expect if atrioventricular septation goes wrong? Write in the chat, you have a minute. Okay, let's talk about the second type of septation. The second type of septation is what we are calling interatrial septation. Interatrial septation is the separation of the primitive atrium. The primitive atrium needs to be separated into the right and the left atrium. So let's take this to be the heart that we have. These ones being the endocardial cushions, which have already formed to separate the primitive atrium from the primitive ventricle. So these two are the right and the left atrioventricular canals, which are the future sites of the right and the left atrioventricular valves. These are the ones I told you it can be completely blocked, then you call that tricuspid atresia or bicuspid atresia, or they can be too narrow, then you call that tricuspid stenosis or bicuspid stenosis. Okay, now I want us to talk about interatrial septation, which means this chamber here, the primitive atrium, need now to be partitioned, need to be separated into two. The first thing that happens is formation of a septum that grows downward from the roof towards the endocardial cushion. That septum is known as the primary septum. We commonly call it septum primum. The septum primum grows down towards the endocardial cushion. Remember, before the formation of that septum, blood will enter like this and flow this way, as well as this way, as we had demonstrated earlier. That means that we usually want blood to be flowing this way as well as flowing this way. Therefore, as this septum forms, we will not allow it to completely block this channel because we want blood to be flowing from here to this side so that it can flow this way. Having said that, this septum, when it's forming, there'll be still that narrow opening. This narrow opening is what we are calling the primary ostium. You can call it ostium primum. But the septum is growing towards the endocardial cushion. And so it will reach a point where the septum has actually reached the endocardial cushion, which means that the primary ostium is now obliterated. By the time the primary ostium is being obliterated, there is something that is forming on the upper part of the primary septum. A hole is appearing on the upper part of the primary septum. That hole that appears on the upper part of the primary septum is known as the secondary ostium. You can call it ostium secundum. The point here is that we cannot afford to completely separate the right and the left atrium during fetal life. It has to be a communication. So by the time the primary ostium is closing, the secondary ostium has already formed. After that, and at the end, I'll ask you why this must happen. After that, we will form another septum. Now look, that septum is thicker compared to the primary septum. That septum is also to the right of the primary septum. We call that septum the septum secundum, or the secondary septum. Secondary septum is thicker than the primary septum. Secondary septum is also to the right of the primary septum. This secondary septum, which is thicker, is designed in such a way that blood can now flow from the right atrium to the left atrium, but cannot flow from the left atrium to the right atrium. It has been designed in that way. You remember we mentioned that the fetal cardiovascular system is designed in such a way that it can shunt blood away from the pulmonary circulation, but also anticipate radical changes at birth. That is what we mean here now. That in fetal life, the pressure here is too high. So blood will flow from the right atrium like that to the left atrium. That channel that blood uses to flow from the right atrium to the left atrium is known as the foramen ovale. So this is the foramen ovale, this one. This foramen ovale allows shunting of blood from the right atrium to the left atrium. Therefore, foramen ovale is one of the fetal shunts that bypasses the pulmonary circulation. If blood flows from the right atrium to the left atrium, that blood has already escaped the pulmonary circulation. So this is one way in which blood escapes the pulmonary circulation. But why must it be designed this way? With the secondary septum being thick and the primary septum being thin, after birth, the pressure here becomes high. When the pressure here becomes high, this valve will fall like this. It will close the foramen ovale. After birth, we don't want blood to flow from the left to the right atrium. It's a radical change that takes place at the time of birth. So that's why the interatrial septum is designed such way. It will allow shunting, yes, but it also anticipates radical changes at birth. Before we go to interventricular septation, I want to ask you a question again, which you'll answer in the poll in the chat. So this question is, if you have abnormal interatrial septation, what kind of anomalies would you expect? If there is abnormal interatrial septation, the process we just talked about, if it goes abnormal, what kind of congenital heart diseases would you expect? So write that in the chat as well for everyone to see. Okay, let's proceed to the third type of septation. The third type of septation is what we're calling interventricular septation, which is the separation of the primitive ventricle into the lower parts of the right and the left ventricle. For you to separate the primitive ventricle, you'll just form a septum. That septum is called the interventricular septum because separating the right and the left ventricle. The septum has two portions. The lower part of the septum is muscular. The upper part of the septum is membranous. That septum is a complete one, so that we need to have complete separation between the right and the left ventricle. That's it for the interventricular septation. So therefore, if the interventricular septation does not go on well, then you'll get what we call ventricular septal defects, VSDs, which are now holes connecting the right and the left ventricle. I want to ask you a question. If you have a hole connecting the right and the left ventricle, how will blood flow in postnatal life? Will blood flow from the right to left, or blood will flow from left to right? Okay, you can write that in the chat. How will blood flow if you have a ventricular septal defect? Ventricular septal defect is commonly abbreviated as VSD. How will blood in a VSD flow? Right to left or left to right? So I can see the campaigns, they are 50-50. Okay. What will determine the direction of blood flow is blood pressure. What will determine direction of blood flow is blood pressure. So I have disabled the chat so that we can continue. Before birth, the pressure in the right ventricle is higher than the pressure in the left ventricle. So before birth, a VSD will allow right to left shunting of blood. But after birth, blood in the left chambers have higher pressures than blood in the right chambers. We said that in the beginning. So after birth, a VSD will allow left to right shunting of blood. And that's the correct response to the question I've asked you. The third, or rather the fourth type of septation is what we call the conotruncal septation. This is basically the splitting of the bulbous cordis. But remember, the bulbous cordis had two components: the truncus arteriosus component as well as the conus arteriosus components. The lower part of the bulbous cordis is what we call the conus arteriosus. That lower part of the bulbous cordis, the conus arteriosus, will be split by a septum into the upper parts of the right and the left ventricles. Those are the smooth parts of the right and the left ventricle. Then the truncus arteriosus, which is the distal component of the bulbous cordis. Truncus arteriosus will be split by a septum as well into the roots of the two great arteries, which means one side the aorta and another side the pulmonary trunk. Now, what do you notice about this septum? You see the left ventricle is usually on the left and the right ventricle is usually on the right. So the septum separating the bulbous cordis, you can call it conotruncal septum or aorticopulmonary septum. What do you notice about this septum? This septum has a spiral outline. And that is why lower down, the pulmonary trunk is on the right and aorta is on the left. Somewhere in the middle, pulmonary trunk is anterior, aorta is posterior. And up here, aorta is on the right, pulmonary trunk is on the left. It is because the aorticopulmonary septum has a spiral outline. And so because of that spiral outline, there are positional changes in the great arteries. Pulmonary trunk is initially from the right side. Remember, it's from the right ventricle anyway. It goes in front and then to the left. And that's why the sound of pulmonary valve is heard on the left, in as much as pulmonary trunk is from the right side. Aorta is initially from the left ventricle. It passes posterior and then to the right. And that's why sound from the aortic valve is heard on the right, in as much as the aorta is from the left ventricle. If you have abnormal conotruncal septation, there are many congenital anomalies that will arise. Some of them may not be compatible with life. We'll be talking about those ones later next week. No, next week, but one. Next week are cats. Good. I want to invite you for questions again.